And we move on to our final speaker of this meeting. He's a returning guest at ARDD. Someone could say that he's like a part of ARDD almost. We can't have ARDD without Vadim. Welcome, Vadim Gladyshev.
Thank you, Morten. I think I've been at all, or maybe missed one conference. ARDD is really the best conference. So I counted how many talks one could hear in this program. I counted 106 plus 2 panels. Yeah, there are some parallel talks which kind of excluded. So let's call these people who listen to all these talks above 100 centenarians. Here's one. Morten is a centenarian, AV people are centenarians. Anybody else? Yeah, amazing. Yeah. These people defied time, defied the clock. But even though there were so many talks at this conference, actually I heard very little on actually the topic that we study. What actually do we study? We study aging. What is it? Nobody gave a definition and really didn't explain, well, what exactly we're targeting. So I'd like to start my talk with actually pointing to this particular issue. So in this sequence of events, starting from damage to functional decline to disease and mortality, to me, aging starts here at the damage. And I define damage in a very broad sense. It's not just molecular damage, but damage that happens at the level of cells, tissues, and also the entire organism. So kind of emergent properties which are mostly excluded from the analysis currently. And eventually, this damage results in functional decline, diseases, and mortality. I would say almost entire longevity biotech focuses on late manifestations of aging, and of course, almost the entire medicine. So, of course, we also do that, although we would really prefer to target the aging itself, which is very difficult because it's unclear exactly how to define, design experiments to address that. We published some papers over the years on this topic, and I will tell you a little bit on kind of ongoing research in the lab. First, we'd like to understand general principles of lifespan control. How exactly do we move from the kind of short-lived state to the longer-lived state? And for this, we study signatures of longevity. We study them across cell types, mammals, and interventions. For example, some cells are short-lived, some longer-lived, some mammals short-lived, some longer-lived, and some organisms shorter-lived, but subjected to interventions, they are longer-lived. What exactly is changed? What are these principles of how we could manipulate the organism at different levels so that it becomes longer-lived? This results in so-called signatures of longevity, which are typically omics patterns, which we develop from this comparative analysis and which we apply in various experiments. In parallel, we study changes with age. This is completely different molecular patterns, because here is the changes which happen during aging, and we do this at the level of clocks, such as epigenetic clocks, transcriptomic clocks, and also causal biomarkers. In the end, we have these actionable biomarkers of aging, which we use to, for example, test interventions and also identify new interventions. And the third component here is rejuvenation. This is something very new in the field, of course, where we could transition potentially from an older state to a younger state. And we've seen many examples, I would say many, but a few examples of rejuvenation. And I would posit that the transition from the younger to older state and from the older to younger are different transitions. So we could not really reverse aging in exactly the same trajectory as we age. However, we could potentially reduce biological age. And so for this, we need to develop its own rejuvenation signatures. And we also try to do that. And a combination of these 3 factors allows us to target aging and also rejuvenation. Currently, at the level of dietary, pharmacological, and genetic interventions, but more and more at the systemic level, where we could perhaps in the future achieve more radical changes in lifespan. So we recently published this paper where we we analyzed gene expression across species. And there have been 41 species of mammals profiled at 3 organs, brain, liver, and kidney, with multiple representatives, so it's a very large kind of omics data set. Species are shown here, and here are the representative genes, 1, 2, 3, in brain, liver, and kidney. And you can see that if a gene is increased in the expression with the increase in lifespan across species, it typically does so across species, across tissues like brain, and so in the liver and the kidney. If it decreases, it also decreases in 3 tissues. It kind of helps us really to develop kind of multi-tissue parameters of longevity. We also published a paper already 4 years ago, this one, where we studied this relationship across interventions. And at that point, we analyzed 17 interventions, but now many more, where we also try to understand principles. Is there like a general patterns, omics patterns that lead to longevity, or there are different? And for example, this is one cluster, we call it calorie restriction cluster, which basically indicates that many interventions, they behave very similarly. They modify organism in a similar way. But there are also other interventions that do it differently. Now we'd like to combine what we analyze across species with what we analyze across interventions and what we analyze across aging. There are 3 different, completely different omics patterns. And this is shown here. So here, shown in blue, we study patterns of longevity across species, like brain across mammals, yeah? Or for example, brain residual across mammals. Here's a kidney across mammals, so kidney residual, liver, and so on. This is the omics patterns that kind of represent of how nature extends lifespan. Nature does something like changes like in the mouse-like species, and the organism becomes longer lived. Yeah? This here, we study aging, like aging in the muscle across mammals, brain across mammals. This is aging of human across tissues, global kind of across tissues and mammals, liver, mouse, rat, and so on, patterns of aging. And here we study interventions. Growth hormone receptor deficiency, calorie restriction, rapamycin, and other kind of interventions. And you can see that they all cluster, like longevity cross-species clusters with each other, aging clusters with each other, and interventions cluster with each other. What is interesting is when we compare these patterns. And the findings are quite surprising. First, if we compare longevity cross-species and longevity cross-interventions, we see no correlation. So whatever we study at the level of species is not how nature extends lifespan. And so I would posit that the potential of what nature does is mostly untapped. We would need to understand better how exactly nature does it and utilize it in our approaches, because we're currently almost here, always here. The second interesting observation is that long-lived species slightly positively correlate with aging. At the level of aging, they are older, not younger. Of course, initially it was puzzling. The way we explained to us this initially puzzling observation is that longer-lived species, they develop adaptations against aging. Yeah? So during aging, of course, damage accumulates, but also organism responds to this damage by activating protective systems. And perhaps long-lived mammals, they also utilize same protective systems. And therefore, it appears that they may be older. Yeah, of course they are not. So, and also we find that there is a negative correlation between aging and interventions, which of course makes sense because interventions, they kind of reverse or can slow down the aging process. This is kind of expected. So then we try to understand that at molecular level. So here again, interventions, and then aging and maximal lifespan, adult body weight, which is life history traits. And all other kind of circles here are molecular processes, key molecular processes that we identify to represent and link these 3 manifestations of lifespan and aging. So for example, mitochondrial translation, you can see, is linked, ribosome, oxidative phosphorylation, DNA repair. There have been many talks at this conference on this topic, but also individual kind of genes and processes. We are at the very beginning to understand this very complex relationship among these processes. But it's very exciting that for the first time we are able to at least begin to understand that. I would also say that the omics patterns that we develop can be directly used to identify new longevity interventions. And here is a figure from old paper where we actually used our initial patterns to scan public data sets and also do a chemical screen. And these are 4 compounds that we identified initially that they would be longevity interventions. And we recently tested this one, the KU compound. We actually found it extends lifespan. We included this in a recent Cell paper. We also found this compound, also this published in collaboration this year, relmindin, also extends lifespan, and we're currently testing some other compounds. We have many compounds. Actually, we have a large currently mouse study where we test many predicted compounds. Hopefully, I will report on this next year. Now I'd like to move from these patterns of lifespan control to age-related changes. And they are often represented by hallmarks of aging. And I took this figure from a nice paper by Dorota Skowronska-Krawczyk, and where she illustrates these hallmarks of aging. And of course, it's a very useful concept, but to me, it's not sufficiently systemic and also not really quantitative. So we often instead use biomarkers of aging, so-called aging clocks, and Horvath clocks, because Steve Horvath is the first one who actually conceived this idea. And I will show you a couple of examples from recent papers that we published this year. In this paper, we studied the effect of severe stress on biological age, as defined by second-generation epigenetic clocks. And we found that biological age is reversibly increased by severe stress. So severe stress somehow transiently changes age. We literally become older, and once the stress is relieved, we kind of normalize age. So Morton, I can see, he's almost sleeping. He is definitely high biological age now because he's under stress. But next week, you will be okay, Morton, don't worry. In the paper, though, we described examples of major surgery, COVID-19. It's not COVID-19, but kind of severe, somebody goes to ventilator in a hospital and then recovers. And also pregnancy. Women in the 3rd trimester they are literally older than early in the pregnancy, and the age recovers postpartum. Also, parabiosis, where we study the young mice which have joint circulation with old mice, and they literally become older. And once we separate them, the age is also normalized. Speaking of parabiosis, we also published this paper recently. This is the work of Bahan Zeng, who's a postdoctoral fellow in the lab, and also Alex Trapp contributed, and others. And this is a collaboration with Jim White at Duke. Here, mice were connected for 3 months, and after 3 months, we separated them and followed for another 2 months, and then later until they die, and we study what happens to them. And interestingly, we found that mice become longer-lived. So once there is a joint circulation between old and young mice literally rejuvenate the old mice. And then at the level of epigenetic clocks, we observe that they are also younger. The effect is very, very strong after 3 months, and it persists after 2 months after they are separated. But later, it's diminished. So we still see the statistically significant difference in lifespan, although the effect is much smaller than the effect at the level of clocks early in life when they've been just separated, for example. So this is, to us, it's a very strong example of rejuvenation. It shows that rejuvenation is possible, and we're actively trying to understand mechanisms of this process. Another example of rejuvenation is we described in a series of papers over the years, and this is rejuvenation during embryogenesis. By applying various clocks, we found that at the time of conception, the age of the zygote is low, very low, but it's not 0. It's slightly above 0. And then during early stages of embryogenesis, we observe this rejuvenation, culminating in the point that we call ground zero, which is the lowest point of lowest biological age. And then age begins to increase. And we think aging, the process of aging, actually begins at that point, which is in mice approximately 7 to 8 days post-conception, and in human it's going to be a 3rd week post-conception. An update to this is that we have another paper which soon will be published in Age and Cell, where we study this in humans also. And we found also, like previously we found this in mice at the level of bulk clocks, single cell clocks. We observed this in frogs. We also observed this in humans. There is a rejuvenation also happen, culminating in ground zero. We also apply other clocks, and we are most excited of this transcriptomic clock that was developed by Alex Tishkovsky. This is a really excellent tool, and currently in the lab we apply both epigenetic and transcriptomic clock because they are kind of orthogonal measures of aging. This is based on more than 3,000 samples. For example, we sequenced many of the samples from the ITP program, interventions that both extend lifespan and don't. This clock really works really well. I will show you just a couple of examples of that. And actually, this is an example where we apply this to single cell data. It also works at the single cell level. So this is the data from Tabula Muris data set. And this is application, for example, you see in the bladder, heart and aorta, kidney. It works, this transcriptomic clock, to predict the age. And here we apply this to rejuvenation upon reprogramming. So on the left here we study OSKM reprogramming by Yamanaka factors of human fibroblasts. And you can see here we study the fibroblasts themselves, and we can see, of course, age-related changes. We can quantify a biological age. But when we reprogram, when cells are reprogrammed to iPS cells, this aging pattern is completely lost. Of course, as expected, iPS cells, they are always young. Doesn't matter, they can proliferate and passage, they would remain young. And here we apply this to partial chemical reprogramming, which is also a very exciting direction being currently pursued by many labs. And this figure from this paper by Wayne Mitchell, actually Wayne is here, hopefully you've seen his poster here, and paper in bioRxiv currently, where we used chemical cocktails of 2 compounds or 7 compounds to reprogram partially fibroblasts, and we could see based on the transcriptomic clock that the biological age of these cells is decreased. And there's another also paper from the lab of David Sinclair. He also mentioned it in his talk, which also shows similar kind of changes. And this is application of the transcriptomic clock to embryonic rejuvenation. I told you that there is during embryogenesis rejuvenation, and in addition to epigenetic clocks, we now confirm that with the transcriptomic clock, you can see this is the zygote, this is ground zero, and this is where aging begins during embryogenesis. It's all good, but now I'd like to tell you about the problems with the clocks. You already heard from several talks that we actually don't really know how the clocks work and what exactly we're measuring. And to illustrate this point, I will show you this image of the same person in the younger and older state. You can see during aging, this person now wears glasses and uses hearing aid, and you can see some other changes, right? Of course, this is not aging per se. This is a response to aging. This person has a poor eyesight and therefore wears glasses. However, it's an age-related change. So if we build a clock, the wearing of glasses is going to be a part of the clock. And of course, at the molecular level, it's exactly what happens. So we need to separate these causal changes. And hopefully you heard the talk by Albert Ying, a talented graduate student in the lab who developed the first causal clock. So first the so-called cause age, and then dam age, which is clock based on damaging changes, as well as adapt age, clock based on adaptive changes exclusively. And you can see both of these clocks, whether based on damage or responses to damage, they can quantify the aging process. Here is, for example, our molecular changes, like byproducts of metabolism, mutations, epigenetic drift. This would be a clock based on that. But adaptive age, this would be like wearing glasses, hearing aid. It also can quantify age, but we cannot use it to test interventions. And the examples of this is shown here. When we reprogram cells from fibroblasts to iPS cells, damage-based clock works to show rejuvenation, but adaptive age clock does not. So we really need to understand better the nature of the clocks and develop better clocks before they can be used in interventions. Here is another problem with these clocks, and this is the work of Andrei Tarkhov, currently in bioRxiv, and the paper is under review. And he studied aging at the level of single cells, epigenetic aging single cells, and also at the bulk level. And of course, we may observe co-regulated changes. If one side changes, then the other changes consistently across cells and across organisms. But also could be stochastic changes, like random, completely random changes. If we analyze both of these, we actually find that there are regulatory changes that we can clearly observe in the data set. And we also can find stochastic changes. So like everything in aging, it's not just damage and adaptation. It's not just stochastic and regulatory responses. And it's not mitochondria, not telomeres, not proteostasis, not liver, not brain. It's a system. And there are very little studies actually on this systemic, holistic view on aging. It's very difficult to develop biomarkers for this process. And so as a result, we have biomarker evolution. We have the ability to quantify various aspects of aging, test longevity interventions, and there are opportunities for major advances, and major advances already have happened. And yet, there are challenges. Many questions remain, and there's a consistent skepticism, as you can see, in the field. So, to address this, we recently organized a consortium called Biomarkers of Aging Consortium. And there have been several studies. This is the first study actually published yesterday in Cell. And these are the authors. 11 of these people gave talks at this meeting. So it's very well represented here. And all of them mentioned clocks, by the way, on biomarkers. And of course, this process will continue because this consortium, but not only this consortium, other groups, they need to develop consensus. Like here in this paper, we try to basically develop consensus. What is biomarker of aging? What is biological age? What is aging, actually? And how do we develop biomarkers? What are types of biomarkers? What are clinical trials that can be applied, and so on? And, of course, the work needs to continue. I would like also to mention this symposium that we will have in December, specifically on that topic, on the topic of biomarkers of aging. This work on the development of this consortium and also symposium is actually led by these three remarkable people, Madi Mokri, Chiara Herzog, and Jesse Paganik. The junior people, like Jesse is a member of my lab, and Madi is a member of my lab, and also Mike Snyder's lab at Stanford, and Chiara is a postdoctoral fellow in Austria, but they're really the leaders of that effort. And of course, many senior people, leaders in the field are here as well. So finally, I'd like to acknowledge people who have been involved in this work. I showed you the work of Jesse and Alex, Bohan, Madi, Albert, and Wayne. And also the previous lab members, in particular Alex, Chaba, and Anastasia, who are here at the conference. They already left the lab, but with new positions, but of course, very happy for them. And also, we have amazing collaborators that all of our studies are done in collaboration. I sometimes maybe forget to mention, but this is really a very collaborative lab. And many sources of funding, which of course we are very grateful for that. So this is kind of last talk, yeah? Kind of final talk, final line, so to speak, yeah? Yeah, with so many kind of talks and meetings and posters and mentions of clocks in talks and bars, cocktails in bars, in bar 7. So I think we kind of defied time and defied the clock. So, and for that, actually, we are very grateful to Morten and Danielle and Alex. I don't see them here, only see. Okay, Danielle is here, and Alex was right there. Oh, he's jumping here, okay. So thank you very much for this amazing time that we have had here, for this. Yeah. This is a perfect conference, and we now can take the information we gained here to go home and work even harder to really defy time and defy the clock. Thank you.